You know, I was observing, I was trying to think what Machiavelli would say about going as the last speaker before the bar, and I was thinking to myself that we could do the genetic experiment as what is our alcohol flush reaction, or we could do the direct experiment and just measure it in the observation after the talk is over. But I'm conscious at the end of the day, and I want to get through the data quickly and talk a little bit about where we're going. And I think some of what I'm going to talk about integrates some of the concepts outlined today. I'm going to start going back and describe that I think the Greeks sort of knew what we were up against, and this is not a new concept. And the question of personalized, and we're going to go through what it means a little bit in my talk, is not necessarily about a technology or an approach, but just a holistic way of thinking about the individual and their treatment. And there are many things in this that I think we have to reflect on and go through. So I'm going to start with Hippocrates and end at Google. So we're going to go through the ages a little bit over the deck today. So why are we talking about this? I think that medicine has operated with a long history of slowly evolving models to deliver dramatic changes and improvements in human health. Many of those came from just public health and sort of public service work, but medicine has dramatically improved things using a series of methods for a long time. And I think that those are about to be challenged by 5 forces that really are going to upend the system in dramatic ways and much faster than people anticipate. So the first is the empowered patient. And this is a group of people that will use new information and tools to hold the system accountable for what they perceive to be failure to use the best information. And perceive is important because it may not always be accurate. But there will be people with resources and brains that are not trained in the system using their data to challenge the assumptions of the system in much more significant ways than have happened in the past. The second is molecular medicine, and I'm going to come back to that at the end because I think it's actually the most important. But of the other 4, what's the doctor's role in this new system? There's a great graph in an IOM report that's a committee that I'm part of that shows the number of data points used in each clinical encounter. And it goes, 3, 4, now it's about 10. And it anticipates the number being hundreds in a few years. I actually think that's way off. I think it's going to be in the thousands. And so what is the doctor's role in that environment as a data integrator, as what has previously been a wonderful heuristics machine to integrate the best solution in an environment where the data is shifting very quickly? We're used to some very traditional ways of generating data. Particularly the drug industry has been a monopoly supplier of their own version of what works and what does not work. And this monopoly relationship is being broken. Today, Kaiser has views on whether drugs work. The National Health Service is measuring their own outcomes. People are beginning to challenge in the real world the trials or the definitive data. And we don't really have a methodology for integrating these disparate sources right now into a holistic understanding. Ongoing reform challenges all of this, and we heard it today about how much we spend. And I don't know if that's a problem or not. I think that we have to think through, if health is something we value, and if we get value for our health, then one might think we want to spend more. And I think the question is not really about, are we trying to lower costs or are we trying to create value for society? And I think we framed this in a way that actually might make us not capture the value we could create if we thought about it in a more effective way as a society. So why molecular medicine? I ran a drug discovery lab for a decade. Every mouse in our lab that received a drug had multiple tissues gene chipped. So we collected 30,000 data points per tissue. We did in situ hybridization confirmation of most of the changes that we thought were interesting. We did RT-PCR and all the genes of interest. And we did this because we could not deal with the variants in our cloned mice to understand the effects of drugs. None of those technologies are used in medicine today. They are all essentially free. They cost less at some level than a standard blood test would occur in a hospital. And I think the transition of going from 10 to 20 to 50 data points to 50,000 is not something we're prepared for, either in our understanding of disease or in our understanding of the consequence of treatment. And I want to talk about some ways of going through that. The proposal jumping to the end, is that the formal methods that we use in clinical research, which sit on their own, when we want to answer a question, we identify two population groups and we test something, should be brought into the clinical environment. And I'll go through that in 1 second, but essentially I want these two systems to be identical. There should be no distinction. In fact, in an ideal world, other than in very rare circumstances where you need to run a causality proof, I don't think there should be trials. There should just be care with new interventions introduced in an effective way. The second part of this is that patient value has been an afterthought in these systems, and it is time for the value to the patient, the well-being and the productivity of the human being, to drive the system. So how do we bring this voice in so that it overrides the considerations for economic or other things that are driving this behavior? So that's the shift that we want to cause, and we're going to talk today about how to do that. So to go back, clinical research, this is a trial that was done in ALS recently that tested lithium carbonate and riluzole in two populations. It used a formal array of data, start, 3 months, 6 months, 9 months, 12, 18 months of data. And it used the double-blind methodology to isolate the two groups and create an artificial experiment to see, all other things being equal, does the treatment modify progression of the disease? And this is how we know most of what we know about drug effects in medicine, that we believe in a confidential way, in a confident way. And the result of this test in ALS was that the patients on drug did significantly better than the patients not on drug. And having worked in ALS or motor neuron disease for a decade, we had never seen data like this. This was very, very exciting. This was a transformative data. Riluzole, the drug that's approved, would barely distinguish in those lines in its effect. Now, what this results in is essentially, and this is, I think, the beginnings of where we need to go, a claim, formally, that an intervention, lithium and riluzole, together produce more efficacy over the risks and tolerability, that theoretically, and it's a functional rating scale, improve the well-being and productivity of the patient for a specific pathology. And this is the equation, and we can measure the value of that, and that is ideally what we're doing. But what I like about this diagram, this circuit diagram, is it formalizes a story. And medicine is too much about stories and not enough about equations. This is an explicit semantic claim. If we make the claim, we can write it in a computable way that it can be integrated across systems. Clinical care, on the other hand, and this is my brother Stephen, who was diagnosed with ALS in 1998, results in a note. CCR is the format, or HL7 we use in the US. And it has some data. They have ALS. By the way, just to give you a reference point, if you are a quadriplegic, it turns out, according to medical record data, you have a 60% chance of remission on your next visit because it's not recorded as a new condition. You have a little bit of lab data. You have a med list that may or may not be accurate, and you have a complaint and then a solution. And this is a good clinical encounter. It's not computable. It cannot be integrated well, and I cannot generate any real understanding of disease from this, and it certainly will not go to help anyone else in the future, like my brother, based on the experiment that they've done, whether Ambien and Detrol made any improvement to his issues. So how do we begin to address this problem? I think the key is in going to the patient. And in going to the patient, you're really looking at what Stephen asked, and this was a question that came out of our work with patients. Given my status, what is the best outcome I can hope to achieve, and how do I get there? And I would suggest that this is actually a great foundation on which to judge medicine's effectiveness. Status is a specific computable context. All of the variables that are meaningful to me, that are variants from normal health, should be recorded in explicit ways. Outcomes are the things I both desire and reduction in my pathology, the ability to achieve those, their goals. And they can be explicit and computable. And the journey is a set of interventions that can be described in, again, a rigorous and effective way so that we know what was done. Stephen's encounter was unsatisfactory to us, even though we had a great doctor. And so we built a EMR that we allowed us to build something for patients. And this is a version of this. I'm going to show it to you in 1 second live. But I want to give you the origin of this. I was running a clinical trial, and I was building a data system to collect the data. And my wife left me because I'm a little bit obsessed with research, which is a trait that's not uncommon for those of us in research. And I was online dating. And I was looking on Match.com, and I was plugging in, I wanted a tall brunette, and I was looking for a certain favorite book. And I found her, and I have 2 wonderful kids with her. But what I realized was that the dating site was a clinical assessment tool that used the same data architecture that the patient-reported outcomes I needed for my patients was in the trial. And if we collected that data and let the patients interact and relate to it, we could build a dating site that allowed patients to interact. So I'm just going to show you live. I'm going to do a demo dangerously. So let's make sure we're still online. Here we go. So there are now 115,000 patients in our dating site. And I can find patients like me. So let's say I have a condition, multiple sclerosis. And I'm somewhere between the age of 35 and 50. I'm male, which would make me a minority in the multiple sclerosis patients. I take Copaxone. And I have depression. There are 38 patients publicly sharing their data that have all of those criteria. There are another 124 patients just like them in the system. If I look at one of these patients, if I hover for a second here, I could see that this person has mild social, mental, and physical disruption, so they're not ideal. They want to be better. Their cognition is impaired. Their speech is normal. Their vision is impaired. And their sensation, they're in pain, and they have walking issues. If I go and look at this patient, and I've never looked at this patient before, you see I have a photo, just like Facebook. I have the clinical assessment of what's going on. And then I have the data. And I can look at whatever length of data I want, so the entire duration of their disease for the last 7 years. And they've been steady in their disability. I can see the scores over time. This is prospective. Since they joined the system, these were retrospective. I have their social, mental, and physical well-being, the drugs they take, more than the drugs they take, why they take the drug. So he takes Cymbalta for depressed mood, and he has side effects, and it's longitudinally stored. He takes Prilosec and has a side effect of dry mouth. I can look at his symptomology, which includes his depression, anxiety, even sexual dysfunction level. And you can see every date and what's going on. And this is public. I'm not logged in. You can go do this on your own. Weight, other biometrics. This is sort of the way we think about what medicine could do. Note that this patient has been viewed 5,226 times. So 5,226 other patients have used this information to help them understand their own disease. So normally we see our doctor 2 hours a year, if we're lucky. The other 9,000 hours we have to live with our condition, and finding someone else like you to manage that is very helpful. If I click on one of these drugs, like Cymbalta, it's going to take me to a summary of the 2,600 patients using Cymbalta, why they use it, fibromyalgia, depressed mood, nerve pain, anxious mood, how well they rate it for that function, so it's better rated for depression than it is for fibromyalgia. You can see the side effect level. And I could click on any of these. Again, if I want to look at people that have decreased libido, well, I can drill to the symptom, which is tell me the list of people, and then the people that are having that as an experience. So this tells you and how many are severe, and all the other variables across the board, and what causes it, the different things that connect to it. We can do some cool things. We integrate, every day we download the entire clinicaltrials.gov database from the US government. And we tell you every new trial that you are eligible for And you can join that. And if you want to filter it, you could just say, I'm only interested in phase 3 drug trials within 25 miles of my house. And it's going to refilter the results and generate, of the 13 trials I'm eligible for, how many are phase 3 drug trials. And there are 6. And if I were in the system, I could say, I tried to enroll, I'm not eligible. I could track that, and we could feed that back to the trial managers. So you get a sense of what the system can do and how we use it. I'm happy to answer questions later and show you what it does. You know, what we really have is a data architecture. That's my brother. Those are all of his symptoms. Those are his treatments for his condition and his symptoms. And they're linked. There's attributions. This is not just state data. This is a data with attributions between it. Purpose, connection. Why does it happen? These are the side effects for his symptoms. Sorry, for the treatment for his symptoms. And imagine if you have this computable data, what you can do. You can say, in the order in which the symptoms matter to me, tell me what is helping and hurting for everything that I am doing. So, excess saliva is a problem my brother hated, because we used to like to go out to the Ritz-Carlton and pour red wine down his feeding tube. And he didn't like drooling, so he treated it with Botox in his salivary glands, hycosamine, and amitriptyline. 2 drugs had the beneficial side effect of dry mouth. 4 of the drugs caused constipation, which we managed with magnesium citrate and CoQ10. But even after having read every drug monograph for the known side effects, I didn't put together, because I didn't have the data in the right format, that 5 of the drugs were contributing to his insomnia. And that causes pain and depression. And without that information, he can't modulate or manage the things that are most important to his quality of life. So, what can we do with this, with these new eyes? Well, we have the clinical trial here for Lyrica, which is a drug approved for fibromyalgia. We have we have our data on Lyrica, smaller numbers, but we're still young. We've only been doing this for a year or so. We have 37% of our patients are experiencing weight gain, 11% of the people in the trial. Now, that's obviously apples and oranges. In fact, the New England Journal of Medicine had a great publication that showed that doctors underreport patient side effects by roughly half for most indications. So that data is not out of line with that report. But what we can do is look at apples and apples and oranges. And you can compare Lyrica to Neurontin, which is the same. Lyrica is the prodrug, the metabolite of Neurontin, and Cymbalta, which is a different class of drug, all indicated for fibromyalgia, though not approved, and compare the signal in our system to the signal in their system. And the signal in our system might be better. It might be really indicating more weight gain than the others. But that's just an opinion. Can we go beyond opinion? Can we drill into the data? And what you can say is, and these are very small n's, because we didn't ask people to enter their weight when they started the drug, that patients on Lyrica gain 10 pounds of weight a year, and patients on Cymbalta do not. So if you're a woman with fibromyalgia, and you are offered 2 drugs of equal efficacy of different classes, and one will cause 10 pounds of weight gain a year, and one will not, which would you prefer? Would you like that information? Because you will not get it in your clinical encounter right now. I think what we're building is a recursive model to create great health engagement. The patients understand their health by entering it, by quantifying it. They find support from others from that experience. They learn how they are different and the same, which results in a better ability to go back to the clinician and have an excellent dialogue in the hours and time you have. And then the cycle repeats. And you get better and better outcomes, I think, when patients are engaged in this way. We've actually measured the benefits. This is data from our epilepsy community. And 18% of epilepsy patients reported fewer visits to the ER on the basis of the things they learned in our system. Now, they also sought specialists. So whether this caused this, or understanding lower side effects kept them more compliant, whatever the variables is, if I had a drug that could reduce ER admissions by 18% in epilepsy, I would be a very wealthy man right now. But the ability to use information to do that is not currently reimbursed in our medical system anywhere. We also had this neat effect where a couple of patients got married, and we're now studying, as again, it was a dating site, what are the side effects of engagement? What are the genetic consequences of patients getting married? We actually have 4 marriages in different diseases, and they're all within disease. So 2 Parkinson's patients, We have an interesting effect. We've measured that the more social ties you have in the system, the more benefit you get from it. Again, that's a coincidental observation, but the more people you claim to be relating to in the dialogue, and we've measured this a bunch of ways, the more you actually learn. So again, group therapy seems to work, and there is a dose-response curve for it. So this comes back to the very exciting study at the beginning, where lithium stops ALS. And I noticed a lot of the dialogue tonight is about how internet companies or these advanced research models can produce really inappropriate behavior on the part of patients. So let me tell you what happens when PNAS publishes a small study with limited power that a drug makes a difference in ALS. 10% of the patients in the world that we knew of took a drug with neurological damage as a potential side effect with their doctor. 10%. So, when we talk about the theoretical harm of learning you have Alzheimer's versus the actual harm of I think it's a good journal, right? Of a journal publishing something that changes behavior in an inappropriate, unthought out way, we have to balance those issues. And in this case, this particular patient, who started taking lithium, by the way, based on an Italian translated conference proceeding, hacked our system. He downloaded all the data from all the patients and started putting a graph on Google that said, this is what's happening to the patients on lithium versus the average control in patients like me. And we panicked. We said, well, that's not going to be predictive. You can't do that. There's too much bias. The model is wrong. But we said we can't leave them hanging, so we had to do it better. So how do we make this data useful? I mean, most people would say it's not a double-blind study, you can't make a claim. So can we do something? Well, we did some research, and I think essentially what we do, by the way, is bias research. That's what we do at our enterprise. So the first thing is, if I match by gender or age or whatever normal criteria we would use, what do I get? And the answer is the patients on lithium, and I love this line, before the start of our clinical study, we evaluated the relative progression rates of the patients. And what I like about that is that I can state that we evaluated something before the start of the study because we had the data. So before the start of the study, the patients on lithium were getting worse faster than the patients who were the controls. Now that makes sense because it's a desperate disease and people that are going faster are more likely to take an experimental treatment. Men go a little faster too, and men are more likely to take experimental treatments as well. But we understood that and we said, okay, well, we can fix this. Let's lock how long you have been sick with how sick you are. So we'll lock both ends of this line and it'll match. So we did that. And this time, on many of the runs, and some of them actually hit borderline significance, the patients in lithium were getting worse slower before the start of the study. Now that was really confusing. We were trying to understand why would that happen. And our physicist who studies the finance markets, we actually took him after he crashed Wall Street, hypothesized the following. Here's two patients, and one would say, wow, that drug is doing something, so that's a good effect. Unless you looked at a background curve that looked like that, in which case if you looked at the actual full vector of the patient's course, it's kind of predicted. So if it's predicted, when you look at the preceding 12 months, you end up with a separation. And in fact, in all of our models, the data crosses perfectly. So we said, how do we do this? So with some fancy math and some fancy graphics and a few physicists, we asked this system, so computer, go and get everyone with the exact same course of disease as Humberto and tell me what happens to them. So it's going to get all the medical records, it's like a file, you have to go get all the things out of the library. And then let's put all the numbers in one context against the background curves of all patients. And then let's build an integrated model that predicts that patient's future based on, say, 3,000 to 4,000 patients of data. And the patient died at the 75th percentile of the controls. I can actually put standard errors on this data. It's kind of scary, powerful, isn't it? I can tell you when you need a wheelchair, when you need a feeding tube. If you do this, you have an automatic control group for each patient matched in the full course of the disease. Now, that does not remove all bias, but it got a lot out. And in fact, in our data set, it created a perfectly matched prior set. And when we looked at the data after the fact, now I'm going to tell you how biased this group is. Out of the 300 patients that started taking lithium, 170 of them met our data density requirements with enough data. Only 78 of those stayed on the drug for a year. Now imagine running a trial where the dropout rate was 60 to 70%. You'd assume that those patients were feeling like the drug was doing something. So that is the most biased group we could pick. And the matching off the data, using the prior matching, was perfect. And in fact, we showed there was absolutely no effect. And here was the power to detect. And here's the original study. So we had about we had the power, 80% power to detect 1/6 of the original claim in PNAS. And we had this data, we published the first 6 months of data 9 months after the original study came out at a meeting, before the first patient enrolled in the 4 clinical trials that have all failed for futility at 6 months at a cost of $25 million that have taken 3 years now. None of these trials have 12 months of data. We're the only ones, because they all stopped because the drug didn't do anything. We published this in Nature Biotech, both as the result and then as a method for thinking about how to improve power and controls. Now, I don't know that people think about this normally as personalized medicine, but it is. It's a prediction about the individual patient's future of a health variable. And I would say that our objective here is to think about how to do that prediction on an individual. This feeds back that I think we can refute, and did, this clinical research claim. But if you think about what we need to do personalized medicine at a complete level, it's really about the network. All interventions against all pathology with the goal of improving well-being and productivity, but they all have to make the same rigor that drugs do. We have to define what they are. We have to understand them. We have to know what their variants are. That means surgeons have to be accountable like drug companies. That means that the things we do and the interventions we do have to be measured. We shouldn't do medicine unless we measure what we do, and I'll get to that at the end. In the US, this market is about $1 trillion out of the $2.5 trillion, which leaves about $1.5 trillion of processing, doctor opinions, views, diagnostics, and other things. I think that there's a very big business here, and so do these people, to think about how you take the errors and efficiencies out and you match the right intervention to the right pathology to maximize well-being and productivity. But we have to make this explicit. There's no more stories. It has to be computable and meaningful. So this is what I think. We had to do this for our study, so I'm making these recommendations. The truth standards of disease are not good enough. Breast cancer is not a sufficient definition. Fibromyalgia is not a sufficient definition. These are not things that, in our experience, when we look into patients, even ALS itself is not a sufficient definition. You need to know more. And in fact, in MS, we have data that shows that the types have zero predictive value for the next stage of the patient. We have very little data on host health. Interestingly, we all have the same labs we use in hospitals, and other than those, and some emerging concierge doctor values, we really don't know much about the molecular health of a patient. And we can take that to a new level. I'm stealing this from Zach Gohaney. I become increasingly frustrated by the inclusion of non-medical events in medical analysis. How much of breast cancer as it's practiced is actually real cancer? If we confound things that are like or match pathology measures without actually really mapping it through, we're never going to get great numbers at the back end. We have to define what disease is. And this one I think is the most important. We think of health as a state, a static state. Are you diagnosed with something or not? If we're lucky, we have a diagnosis date, but most medical records don't. We have to think of health as a vector. Both the health itself, how functional are you from birth to death, by the way, and the sum is 0. And then, how do you deviate from that on every measure that's meaningful in a disease? And this has to have an origin and the magnitude of the variants that are important to the patient. And we have to know that. And if you think of it as a vector, you begin to understand what you don't know in between the data points, which is, I think, where we fail in our static diagnostic versions. And this is why it's going to get a lot worse. This is just one of hundreds and hundreds of studies that show networks of disease at the genetic level. And whether or not leukemia ends up being something that we define by pathology or by these sub-relationships of networks of genetic effects, I don't know, but I know that whatever definition we have now is wrong and will change dramatically and rapidly. And in that environment, we might need to think about doing things in a new way. Our concept of disease, most of what we saw today, was an association of a single or a few genes with a condition. And we've learned so much from that. I spent 10 years studying ALS on the basis of a discovery that SOD1 was a genetic form of ALS, and it is one of the most powerful tools ever invented. And we should not in any way underestimate the impact of that and what it can do to change human health. But it's not what we need to get where we need to go. Obviously, there's technologies that look at protein and RNA, and we've used all of these at the single level, all of molecular biology is these associations, and correlating them with what the definition of disease, some definition that a doctor said, but that's not what we need. We need to measure, and we saw great data today, de novo, genetics, the phenomics, most importantly. Because it isn't just absence or presence, it's severity and duration and the relationships that can't be gotten without this. The change in DNA over time, the change in the RNA and the change in the protein. And if we take all of this data and integrate it into holistic models, we'll develop compression algorithms and grammar and tools to make this more effective. We'll stop thinking of health as these diagnostic codes and start thinking of health as this holistic integration of a set of vectors that we can monitor, that we want to optimize, so that we can pay to be healthy. It's a circle that can be virtuous if in every time we give someone something, we measure everything and we measure what happens to them, which we don't do now. And the vision is that someday a patient who has a pathology and a set of goals and dreams that are heading downhill because their problem is getting worse, goes and sees a doctor who consults a system that predicts exactly what will happen to the patient without the intervention and to the patient with the intervention. And you know what? The model will be wrong. But every chance we do that, we can learn why it's wrong and how to make it better. And I think that's the shift that we go from when we think about this isolated clinical research discovery system and this clinical care system. So I got 4 conclusions and then I'm done. Each clinical encounter is a unique experiment that currently is not recorded and does not mean, and I mean computably recorded, obviously we write this stuff down, and does not meaningfully advance our understanding of disease. I assert that that is amoral. That is police tracking criminals without looking at integrating the crimes. That is witnessing a crime and being unwilling to report it. If a patient that you see has an adverse event, and you do not give that information to someone else that can use it, you are responsible for the future adverse events of that drug, no more so than if you witnessed a crime, no less so. We should not pay for health care that does not benefit society, period. The individual need does not overwhelm the society's need, and we have to figure out methodologies of doing that. And again, we should not pay for health care where the data cannot benefit everyone. We need to change our measurements to the true standards of disease and the dynamics of human health. They all need dramatic upgrades, and they need institutional level investment, not one-off models and the little stuff we do now and the way we do investigator work. We need to upgrade at a significant level. That means George needs money. That means that these big labs that do this well need real resources. We're going to need immense longitudinal data sets. We don't know what disease is. We don't know what human physiology is. We don't know what pathology is. And we don't know what the molecular state of the human machine is. And every definition we think we know will change in the next 5 years. And if we don't build a system that has massive amounts of integrated data, we have no hope of cutting through it. And if we do that and integrate the clinical discovery information system into the clinical care environment, we'll build what we're all looking for, which is an effective learning health system that instead of waiting 3 years for some confirmatory study to prove that lithium doesn't work, knows it not in 9 months like we did, but in 3 or 6 months after the first few patients tried it, for everyone. And that's what I think we should build. Thank you.
Thank you, Dr. Heyman, for this in part provocative.
Well, my dad's the doctor, so I'm an engineer. So
Yeah.
I never went to school to stop learning to think.
Okay, questions, comments? Have you been too provocative? Everyone agrees that Michael?
I'll follow your orders. Won't you need the classical clinical trials just for new hypotheses, for new drug treatments? It's great to have this PNAS paper, but and you can rapidly disprove it. But if anybody just randomly starts taking non-standard stuff, you'll always have n equal 1, right? So you need to put your 150 people together on one particular trial, one particular treatment, and see what happens. No?
I think there's two steps to that problem. I mean, do I think in the near future that a regulatory agency should accept a non-double-blinded claim for reimbursed on-patent medicine? No. I don't think that's going to happen. I don't think that's realistic. But I think that phase 4s, which are the ability to observe a drug's effect in the real world, either in a comparative effectiveness framework or in validation of the original study, that this method is actually significantly more power than the methods we're using now. Because it really is about ensuring that the clinical environment is collecting the data at the same or necessary level of rigor to make that equation. I want to touch on the hypothesis thing too. Physics a long time ago divorced the data collection from the theory. And, to my mind, the great theorists in biology are often not the great experimentalists, and the great experimentalists are often not the great theorists. And it would be great to think about letting someone that is good at quality, say manufacturing engineers, define how we build large-scale data sets that drive, the massive improvement in quality we've seen in every consumer good in the last 40 years. Let them define the data set, and then let the hypothesis generators in an open architecture like this, and then the PGP and all the integrating connected things, drill their hypotheses and discover the diamonds. And that's a different kind of way of thinking about biology research that I think is much more potentially effective than the way we do it now.
I think most of us, I certainly would profoundly agree with the points you're making. However, I wondered if you had a perspective on why, for instance, a 10-year initiative by the NHS in the UK, costing hundreds of millions, has completely failed to get any kind of usable integrated electronic data. And I also wondered why, for instance, I know a little bit more about United, why the kind of information you find in healthcare providers in the US is devoid of much of the real information that you would like. And so my question is, what needs to happen to allow those kinds of organizations, healthcare providers, actually to implement an effective thing? Because clearly money and time isn't enough. And my second question, which relates to that, is maybe we should forget completely about the healthcare providers and use PatientsLikeMe to generate the data. That was a question. The question was, why can't they do it? Why can't the NHS? Why can't United?
We're in fairly deep discussions with the NHS. I think the answer to your first question is the most important one. Why has it not worked? We're really talking about building an information market. And information markets need customers that care. And the problem with every effort built to date is that the people that need the information at the end, the users, are disconnected from being able to be accountable for making the system operate level at a detailed point in time. And I think what makes our system work, and it is, by the way, this is such a pilot early, this is a glimmer of what's possible in some easy spaces, not a suggestion that we can do all of medicine right now. But I think what makes it work is that our patients make the system accountable. If the doctors won't give them the data, they fire the doctors. If 10% of our patients told us they fired their doctor based on our system. And I think that What you need is the local accountability for the input to be meaningful and the feedback loop to be fast. And so the patients are the key. And that's the same thing I would say to United, which I do not think will succeed because their data is not particularly useful and has massive amounts of bias and blinding in it. But if I take United's data and the patients and I merge those, suddenly we're really creating value.
Yeah, thank you very much for your presentation. I must admit it really freaks me out. I do not know if in a positive or in a negative way, but it freaks me definitely out. And my question just goes in the direction of the always asked question about data security. What do you do about the patients who want to share their data but who want to stay anonymous? And what about data manipulation? I mean, what presents a pharma company who has a genuine interest in keeping their drugs on the market, just creating hundreds or thousands of profiles who report a positive effect of the drugs? How can you sort them out? Because you've shown it's free, everybody can join, it's open.
Well, there are three questions: the freak out response, and then the pharma companies. And the first one again, give me the statement one more time. First question.
The first was about data security.
Oh, yeah, okay. So you cannot be anonymous in our system. So if you want to be anonymous, tough luck. Go to some doctor that's not going to keep track of your data. Now that will change, but our current social contract is we are building a commons to change the world, and you can come and join us, and those are the terms. We will build private layers eventually, but we do not have any now. And privacy is very, very hard with this kind of data planes. I mean, there's nothing here that couldn't be identified. With regard to the drug company question, we have a bunch of checks on that. You know, we use some of the same tools that credit card fraud and people like that use. It's very hard to game sophisticated data models over time. So it's just hard to lie over time. So single data ratings are not very useful, and they're not weighted very highly. We weight data to the people that give us longitudinal data. But the second one, actually, if you had to pick an industry, so the reason drug companies have been Pfizer's $3.2 billion fine was because the sales rep that cheated got the benefit of cheating. They got paid more if they lied and the kind of stuff that they did. I want you to imagine the fine against a drug company for posing as a patient and entering our system and faking data. Because what they got fined $3.2 billion for was handing doctors papers that were published in peer-reviewed journals that were not on the indication of the drug. So if you look at the magnitude of those two different things, it's not happening. I just don't see it. And secondly, with the numbers we have, I just don't think it's going to make that much difference. And the freak out thing, I get it. My comment on that would be, many people were uncomfortable when the Bible was translated from Latin into languages that the people could understand.
Okay.